And it's my pleasure to introduce the Associate Director of the Faraday Institute, Professor Keith Fox. Keith works 50% of his time for Faraday here in Cambridge, and the other 50% he's a professor in Southampton. He's Professor of Biochemistry and has been head of the Centre for Biological Sciences there. And he's been there since 1987, so you've got a long track record there. His background is in biochemistry and in genetics, and he's going to talk to us about some of the challenging issues that come up in the relation of genetics and bioethics. When you're mic'd up, Keith, we're ready for you.
Does that sound working? Yeah. Working. Good, thank you. Right, so thank you for staying around. I've got a small topic this afternoon, the whole of genetics and the whole of bioethics. So I've asked Hilary that when it comes to 45 minutes is up, just to cut. So if I stop mid-sentence, it's because there's just too much to talk about. Genetics and bioethics. What we're really talking about at the centre of this is this wonderful molecule. It happens to be the molecule that I spend most of my life researching. I'm a DNA biochemist, and it happens to, if you look carefully, it's the structure of my tie as well. It is an icon of our age, almost. You don't have to be a scientist to recognise that as DNA. I know what DNA does, but it's the sort of thing that the man or woman on the Clapham omnibus could identify that as a molecular structure. As a biochemist, I'm proud that at least one molecule has reached the public's imagination. So much so that you can find it almost anywhere. So you find DNA on postage stamps. It's to do with marketing. How that fits in, I do not know. You can even buy DNA perfume, and it's DNA for him and DNA for her. It has nothing to do with DNA at all. It just comes in a helical bottle. And it happens to be a triple helical bottle as well, not a double helical. So it's something that people are familiar with, but what actually does it do? Where does it come from? DNA was discovered itself back in the late 19th century by Miescher in Switzerland. Its structure was determined by Watson and Crick in 1953, who came out with these immortal words, supposedly, "We have discovered the secret of life." Well, I'm talking to theologians, and I think you would agree that there's a little bit more to the secret of life than DNA, but they had discovered something profound in the way in which DNA was replicated, about how properties of life were passed on physically. Remind us what does actually DNA do, and apologies for those of you who come from a very scientific background. And the answer is actually twofold. It does everything and it does nothing. DNA by itself in a bottle does nothing. It is just a white fluffy powder that looks rather like a tissue paper that's gone through the washing machine and been dried out and ironed at the end of it all. It is, however, in the context of a cell, extremely important. It does everything within the cell. It's the repository of our genetic information. It's copied to make more copies of DNA. It's transcribed and translated eventually to make the proteins, which are the workhorses for the cell that do all the metabolism or the structure. And it goes to the intermediate called RNA, which is very similar to DNA. And it's a molecule that, when it's expressed, different genes are active in different tissues at different times. So it's tightly regulated. And it's what defines some of the differences between you and me, as we've got blue eyes or brown eyes, or black hair or white hair in my case. Defines many of the things that make us up. DNA is universal across biology. All living organisms, whether we're talking about bacteria, plants, animals, humans, contain DNA as their genetic material. And it's the sequence of those four bases, A, C, G, and T, that you may be familiar with, will vary from one organism to another, but the chemistry of it is identical. And that means that it's possible to transfer that genetic information from one organism to another one. To take a piece of DNA from a bacterium and stitch it into a plant, or to take a piece of human DNA and stitch it into a bacterium. We'll come back to that later on. The very fact that it's common to all life gives us the ability to move it around. And that ability brings with it a few bioethical issues, just one or two. Questions about the human genome, about human reproduction, about the sorts of things that we learn from the human genome and the applications from it, about genetic technology, about human health and human disease. It impacts on things about biodiversity in the environment. It impacts on what we know about animals, and so we could go on and on with lots and lots of questions, that bioethical questions that arise partially from our knowledge of DNA. So where are we going today? Well, I want to spend a little bit of time firstly thinking about how we make ethical decisions. I feel very conscious of talking to theologians and daring at least to tell you how I think some of the processes we go through to make ethical decisions. We'll look at a little bit of genetic modification. I think that's rather a tired argument now, so we'll spend a little bit of time on that. We'll look at some human genetics. We'll ask whether our characteristics such that we can blame our genes. We'll touch a little bit on epigenetics, and by the time we get to the bottom there, Hilary will have cut, and it'll be time for me to stop. So how do we make ethical decisions? For many people, we just simply muddle through. It's on the spur of the moment, the pub bore, what takes my fancy on the moment. Sometimes, and this I think is important when we're looking at some bioethical things, is we make decisions based on what we might call a yuck factor or a wow factor. The wow factor is this is just so exciting we have to do it. What would happen if we could somehow, because we know its genomic sequence, if we could resurrect a Neanderthal? That would be really exciting, wouldn't it? Well, yes, scientifically it might be, but I'm not sure we should do that. The yuck factor. Some of you may be as old as me to remember the public reaction for the first heart transplant. How could you possibly walk around with somebody else's heart ticking inside you? Yuck, it's just not natural. But now it's, well, not that it's routine, but it's a much more common process. So beware of, if you like, the gut instinct for making ethical decisions. There's what might be called deontological processes, those that adhere to duty or moral rules. And maybe talking to Christians, that may come high on the agenda, as to what do the scriptures say? What is God's rule in something? How should I obey that? Coupled closely to it are rights-based issues, human rights, whatever that might be, the right to health and happiness in the American Constitution, rights-based ethics. It might be called teleological, consequentialist or utilitarian ethics, focusing on the consequences that any action might have. Will it have a good benefit or a bad benefit? Is there a risk associated with it? We might take ethical decisions based on natural law. What's going to have the maximum impact on human flourishing? And Sam Harris, atheist, with his book The Moral Landscape, is probably one of the most recent exponents of that. And again, for those who are Christians, the virtue-based ethic is actually important. Developing good character traits, the fruit of the Spirit, kindness, generosity, acting in a way in which a virtuous person, the person of Jesus Christ, would have done in any situation. And how do we apply those things to biology and to our knowledge of, say, molecular biology? And it's not obvious. The scriptures, unfortunately, don't say anything about DNA or stem cells. So we have to take other principles and apply them and put them within those general principles. We can learn something from the way in which medics apply ethical principles. That of doing good and not doing harm. Now those two, sometimes they balance each other out. In order to benefit the patient, you have to at least temporarily inflict pain upon them. That's what my dentist tells me all the time. Autonomy. The patient has to have some choice, some freedom within the process to choose what to do, and it must be an outcome that is just and fair and not in any way biased. I am now somehow alarmed at some statements from scientists. Now, Steve Jones is somebody who's a geneticist at University College London I admire greatly. But I am slightly concerned about this statement from him about ethical principles, particularly applied to medicine but genetics as well. When it comes to medical research, he says, any medical technology that works is very quickly accepted by the public. Ethicists may not like it, scientists may not like it, but the public, if they believe it works, they will accept it and legislation will always follow. So a kind of utilitarian approach. Ethics has always followed science, it has never led it, and I don't see any reason why genetics is going to be any different. Ethicists would love to tell geneticists what to do, but I'm afraid that geneticists are not going to listen. And there was an article recently written by Steven Pinker, effectively telling also ethicists get out of the way and make space for science. So there's not always a welcome audience for those who want to bring ethical principles into how we apply our science. As Christians, how is that going to affect us? As humans, we acknowledge that we're made in the image of God. We might debate for a long time as to exactly what that means. Our genomes are 98% similar to those of chimpanzees, but that does not mean that chimpanzees are 98% made in the image of God. It may be a 2% difference in genome, but what a massive change that has in physiology and character. So we need to respect our image of Godness that we have. We also need to respect the created order itself, where we're told to rule over, and that obviously doesn't mean in any domineering way, it means to take care of the creation. So anything that we do biologically has to at least have underpinning it the fact that our responsibility is to look after the world that's out there. We'll be hearing more about those in talks, I think, tomorrow and on Friday. But what sort of issues are raised within this? Are we playing at God as we manipulate genomes? Questions actually is about what is natural, what is risk, what does it mean, what is a species? What are species boundaries? Do they matter? What does it mean to be human? Who owns this biological information? And if we're going to be talking about animals, do they have any rights? Or what sort of rights are they? And how do they compare with human rights? Let's look at a few of those phrases. Playing at God is a phrase that you'll often find out there in the media about how we push the boundaries too far and now we're beginning to play at God. And I don't like that phrase for all sorts of reasons. Firstly, it suggests that scientists are playing. Science is great fun. It is intellectually stimulating. It's what gets many of us out of bed in the morning. But it is not playing. It is serious stuff. In the first talk today, it was touched on the creativity of God. If we're made in the image of God, then we should be being creative. If we're looking after the creation, we need to find the best way to do that. But the fact that we can be creative, and the fact we can do something, doesn't necessarily mean that we should do it. There are limits. Ask the question, what is natural? The things that we eat, the organisms that we see around us, particularly in the plant world, are very far from natural. Over centuries of breeding of them and selecting, we've taken what may be primitive species and developed them into something that is high producing. So look at the example on the screen there, primitive popcorn. On the left, you would not recognise that, but it seems to be the ancestor of what we use today as corn. Pigs, very high meat producers. A single mutation seems to have brought about something that has a greater muscle mass, greater meat producers. And what might be shocking to some people is the cereals that we grow, where they originated from. Many of the people who oppose genetic modification will act as though the wheats that we grow, the barleys and all the rest of that, are natural species. Well, no, they're not. In fact, a vast majority of them were obtained by mutagenesis, not targeted mutagenesis, where we're deliberately manipulating the genome in special, specific ways, which is what genetic modification does, but actually ones in the 1940s and 1950s obtained by taking crops and mutating them randomly with gamma rays, chemical mutagens, and X-rays, and taking the seed from that and seeing what gave the best producer. I don't know which I'd rather have my cereal mutated by, genetic means or random radiation chemical means. You can decide. Genetic modification has a lot of applications, many of which we benefit from. GM crops we can argue about, we can discuss for a long time as whether they're good or bad, but they're devised with benefits in mind. Whether it's golden rice, a rice that's designed that has high levels of production of vitamin A that overcomes that deficiency in certain parts of the world. Crops that are resistant to drought or to pesticides. Pharmaceuticals, many of them produced through GM means now, whether it's insulin, no longer extracted from cadavers or extracted from other animals, but grown in bacteria, a human hormone. Herceptin is one of the drugs used for treating certain kinds of breast cancer, is made by genetic modification. You may have come across in the Ebola crisis ZMapp as one of the things in the media, the antibody that was used for treating some patients. ZMapp was made by a virus injected into tobacco leaves, hence the reason why it was rather difficult to produce. So there are lots of benefits from GM applications. However, if you look at the media, genetic modification is something that is often not highly favoured. And I think sometimes we need to be careful about the phrases that we use and whether we bias things. Certain parts of the press will talk about frankenfoods, genetic pollution. Even the word genetics sometimes isn't very helpful. Genetic engineering, no disrespect to any engineers, but engineering they seem to be cowled and calculated and efficient and to do with utility rather than necessarily people. The phrase, and again was touched on in the first talk today, three-parent embryos. Well, no, they're not three-parent embryos. It's mitochondrial transplantation, 2.000001% parents. So we need to be careful about the phrases that we use. Crossing the species barrier. Some people would say, well, God made organisms according to their kind, and that we shouldn't therefore be moving things around between organisms, that define what the Creator has done. But I think you'll have heard from, appreciated from Dennis's talk previously, that defining species is not that easy. There is a continuum almost of biological organisms. There aren't exactly kinds in that sense. Yes, kinds in the sense that sheep give birth to sheep and cows give birth to cows. But the DNA content of those is very similar. Risk is a phrase that frequently comes up when we talk about genetic modification, especially again with plants and crops. I rather like the fact that somebody pointed out that actually the risk to our environment is greater from garden centres than it is from genetic modification, as species from across the globe are brought into places where they ordinarily shouldn't be. Those of you who tried to buy houses recently may have come across Japanese knotweed, which undermines foundations. It shouldn't be growing here. And that is a greater threat to some of our environment than many crops. Opinions we're maybe working against. I'm told that 43% of people in this country think that GM food is very or fairly harmful. And in the EU, an even greater percentage think that it's dangerous, compared with only 4% who think that natural herbal produces are very unsafe. I lecture in pharmacology as well, and I will tell you a little bit more about that as an aside afterwards if you want to. And it's largely the perception of the public on this is influenced very greatly by the organic lobby. Not that the organic food industry is wrong, but they have a very great influence. So it's a little bit on GM, and maybe some rather biased views, you may say. Our understanding of genetics, though, has come a long way since the sequencing of the human genome. Arguably the greatest, I think, thing that humans have done in the past 20 or 30 years was to sequence the 3 billion base pairs of the human genome, first released in 2000 and then several updates since then. Interesting, when that was released, the sort of statements that came out from this. And here is Bill Clinton alongside Francis Collins, who was director of the Human Genome Project, now director of the NIH. And the word God comes into their language. That's not unusual for Francis Collins. He's a committed evangelical Christian, and you may have read some of his books. But the phrase, today we are learning the language in which God created life. We're gaining ever more awe for the complexity, the beauty, the wonder of God's most divine and sacred gift. That's Bill Clinton. And Francis Collins saying, it's humbling for me and awe-inspiring to realise that we've caught the first glimpse of our own instruction book, previously known only to God. A wonderful achievement of science and technology. And at the same time, various releases saying that we've got to be careful about how we use this. The genetic information must never be used to stigmatise or to discriminate against any individual or group. As we'll see in a moment, there is a real danger that can happen in some instances. What can we do with that genetic information, that 3 billion base pairs of data? Well, the first genome to be sequenced cost several $billion. Subsequently, the cost has come down as new technologies have invented. It's not quite down to $1,000 a genome, but it's getting pretty close to it now. But as the sequences of our genomes are known, and we learn about some of the mutations that lie lurking in each of our genomes, and our susceptibilities maybe to various diseases. Who should know that? Should you know it yourself? Should your family know that? Should your GP know it? Should your insurers know it, or your employer? Think about the ramifications of each one of those. If you suffer from, if you have within your genome a potential debilitating disease, should you tell your children, who maybe 30 years later might possibly display some genetic disease? Whether it's 30 years then of worry, possibly. Should you tell your employer that you have a gene that means that you're likely to suffer from heart disease? Should you tell your insurers? You can see the sort of complexities that come into that. I'm not going to give any answers, but these are the sorts of things that people will have to wrestle with. It can be used too, because you can sequence on a single cell, it can be used for diagnosis of genetic conditions. Postnatal, prenatal, pre-implantation even. And some of those have consequences for ethics. Postnatal, like the child is already born, and you'll find out what diseases it might carry. Prenatal, so screening of the fetus in utero, that may warn the parents of what's about to come, but often that's done with a view to the prospect of abortion. And pre-implantation, all right, it's an embryo that hasn't been implanted, and we'll come back and think a little bit about those in a moment. It does actually raise questions about human life and when it begins and what it means to be human. So in the genetic screening, there are various ways in which it can be done. Tay-Sachs syndrome is a genetically inherited disease that's particularly prevalent amongst Ashkenazi Jews. And they have an organisation called the Generation of the Righteous, as it's called. It's a recessive gene, so you need both copies of it to display that disease. So if both of your parents have that recessive gene, they will be perfectly healthy, but there's a 1 in 4 chance that an offspring from that will have the Tay-Sachs syndrome. So they have societies where dating couples can exchange information and decide whether to continue with a relationship before any offspring might even be thought about. And there's a similar sort of thing amongst thalassemia, which is particularly prevalent in Cyprus in the Greek Orthodox community. I'll skip that. There are a large number of genetic diseases that are characterized by simple changes in the genetic code. And here are just a few examples of them. Some of them you might be familiar with. Some are very long names. So we've got muscular dystrophy across here, Crohn's disease. Phenylketonuria is one that's tested for at childbirth. Porphyria, the madness of King George, is what he suffered from. So a whole long list of them, and it could go on way beyond this lot. And a number of genetic diseases, conditions are tested for at birth. 6 by the pinprick test, and 3 other ones with various various incidents subsequently as well. Many genetic diseases come from point mutations. It is clear, if you have that genomic difference, you will get the disease. Whether we're talking about cystic fibrosis, there are some rearrangements in DNA that take place that lead to various cancers and leukemias and lymphomas, and there are expansions of some parts of the genome where bits of it are repeated time after time after time. So instead of it reading the cat ate the fat rat, as it ends up with the cat ate the fat, and the genome stutters. And there's a number of diseases that come from that. Friedreich's ataxia, Huntington's disease, and some forms of motor neuron disease do that as well. And so by knowing these sorts of things, we can start to benefit medicine, personalised medicines, things that are best for your genome. You know that each of us reacts to various drugs in different ways, and that's because our genomes are different, and often because we have different enzymes in our livers for metabolizing them. If you know which enzymes a person's got, you know which drugs to administer them. That's all sorts of benefits in terms of reduced toxicity, reduced cost, improved safety. DNA screening we've already touched on. There's a question about what makes me me. There's me. Here are 6 male generations of my antecedents, my ancestors. I've got white hair. That's actually my mother's fault. 5 of those wear glasses. The one at the top left, my son, doesn't. What other things were genetic on them? 5 of those generations were born in Bristol. 1 was actually born here in Cambridge. Some things are genetic. The bottom 2 wear bow ties, the middle tie wear neckties, and the top 2 don't wear any ties at all. That's clearly not genetic. What makes me me? In terms of, they're all scientists stroke engineers. The appearance, their character, the place where the environment they're brought up. Those of you who travel regularly to Cambridge on the train will see adverts for an organisation called 23andMe, where you can send little swabs of cheek cells off to them, and they will sample your DNA and better tell you what mutations lie hidden within that DNA. What you then do with that information is another question. So can I blame my genes? Have I got the awful character that I have as a result of my father and my mother, or is other factors that affect that? If you read the media, you can think everything is in DNA. It's a word that is misused all the time. Please, when you preach sermons, do not misuse it. And a number of times I hear preachers say, it's in their DNA to do something. Well, no, it's not. It just means that it's their character to do it, as though it's predetermined, there's no escape from it. This is thanks to Dennis Alexander, who collected these. The sorts of things the media portray, a form of determinism. Love cheating, gangsters, meanness, hormone levels, political behaviour, cigarettes, gluttony, liberalness, even to the extent of somebody wrote to The Guardian suggesting that there was a geneticism gene, a gene that predisposes some people to think that behaviour is determined by genes. And it was Dennis who pointed that out to me. Are we really determined entirely by our genes? James Watson, one of the discoverers of the structure of DNA, says this: we used to think that our fate was in the stars, now we know in large measure our fate is in our genes. Is that really the case? Are we indulging in a kind of genetic reductionism, a kind of genes for us fallacy, almost making our DNA, that polymer, that chemical, into a kind of god? dictates what we do that we shouldn't play with? Are we more than just the sum of our genes? And do we at risk of subscribing to what we might call a form of genetic fatalism? And so you see these sorts of things appearing in unusual bits of literature. So in the International Socialist Review, here's an example. The question, is biology destiny? Have we really got any choice in what we do? Are we determined by our genetic makeup? And Nature, a very respectable scientific magazine, it's more than a magazine, a journal, occasionally makes statements that I think are inappropriate. So this is taken from a Nature article in 2012. An increasing number of studies suggest that biology can exert a significant influence on political beliefs and behaviours. Genes could exert a pull on attitudes concerning topics such as abortion, immigration, the death penalty, and pacifism. Way over the top as a statement. Exert a significant influence. Exert maybe have a minor effect, possibly. But this is the sort of language that slips through and is out there. And along with that, then it's difficult to change someone's minds about political issues because their reactions are rooted deep in their physiology. If everything's determined by their genes, don't try and change their opinions. Don't try and convert people. It's all in their genes anyway, whether they're going to believe in God. Now, we don't believe that, do we? And the thing to realise that those who go down that line of genetic determinism tend to indulge in more conservative political ideologies. It influences the ways in which they act. Because if human nature is fixed by our genes, then we can't change society. The problem doesn't lie in the structure of society, but in individuals who make up that society, and the problem with their genes. So we shouldn't be trying to change social structures. Maybe we should indulge in a form of eugenics to eliminate the bad genetic pool. What are their genes for? I've seen some of those on that list. Are some people genetically predisposed to be nasty? Are there genes for criminality? Gentlemen, I'm sorry, but there is a link genetically, and that's called the Y chromosome, which all of you gentlemen have and none of you ladies have. 95%, or over 90% of the inhabitants of jails across the globe are males. Right? So there is a link. But on the other hand, 99.99% of males are not in jails. So it's not a predetermined, because you're a bloke, we should lock you up, because you're nasty. There clearly is some form of link. People have, if you look through some of the older literature, suggested there is a link, a clear link between the Y chromosome. A small number of males, and it's not that small, about 1 in every 1,000 men actually have an extra copy of the Y chromosome. They're XYY instead of just XY. And in most of those cases, they're totally unaware throughout the whole of their life that is the case. Despite earlier claims, the evidence is that there's no greater incidence of criminality in those who have an extra copy of the Y chromosome than those who just have the standard XY. What about other genes for bad behaviour and aggressive behaviour? We all know that our hormones influence the ways in which we behave. Noradrenaline is the hormone that's to do with fight or flight. There are enzymes that break down noradrenaline to restrict its level, to make sure it's cleared out the way quickly when it's used as a transmitter at neurons. The enzyme monoamine oxidase. If you have a deficiency in that, or a low level of that, then the noradrenaline will stay around longer, and we know from experiments with animals that makes them a little bit more aggressive. So there are more than one different form of monoamine oxidase A out there, and the evidence is that those, or the suggestion is that that's been called the warrior gene. Those who have the low activity of that enzyme are more likely to be aggressive. However, a study that was done in Dunedin in New Zealand compared the incidence of violence and aggression, looked at the background, the environment in which those people were brought up. And actually, those who had that weaker version of the gene and were brought up in an abusive background were more likely to be aggressive. But those who had that gene and brought up in a perfectly normal or loving environment, there was no difference between whether they had the weak or the strong form of that gene. So the gene does not determine the way that we behave, it may affect that. Genes don't automatically turn their owners into killers. There are many people with this form of genetic baggage who have never transgressed. In fact, something like about 30% of the Caucasian population have the weak variant of that particular gene and live perfectly normal, civilized, ethical lives. Let me give you a case and see how you respond to it. A case of somebody who taunted a Muslim for wearing eye makeup. And this guy responded to that taunting by stabbing this person to death. He was jailed for 9 years. There was no doubt that he was guilty. The court heard he had a serious history of psychiatric illness. He appealed against the sentence, and in that appeal, the defence lawyers asked for his sentence to be commuted and for some of his genetic background to be taken into consideration. And it was discovered he had various genetic abnormalities, including the weaker form of that gene monoamine oxidase. And in the appeal report, the defence lawyers pleaded that his genes would make him more prone to behaving violently if provoked. What would you do with that information? Would you give him a lighter sentence because he couldn't help it? Would you give him a tougher sentence because, well, he was genetically determined to be bad anyway, so we better lock that person away and throw away the key? You could use the same genetic evidence to argue for tougher or lighter sentences. Could we go down a line where people are actually guilty before they've even committed a crime on the basis of their genetics? If you know that somebody was a genetic risk of misbehaving, would you be blameworthy if you failed to disclose that information? What happened, and it received quite a lot of criticisms in the scientific press, is on the basis of the genetic test, the judge docked a further year off of the defendant's genes, arguing it would make him particularly aggressive in stressful situations, and saying he found the genetic evidence compelling. There was an outcry against that in the scientific literature, because this it implies there's a form of genetic determinism to behaviour, which is not so real. So can I blame my genes? Well, genes may affect our potential, but we still remain responsible for our actions. They may limit us in some ways, but as Christians we know that we are responsible and accountable, each of us, for the things that we do. Moreover, our environment affects us a lot. And this whole area that was touched on earlier of epigenetics, where the things that we do, the things that we experience actually modify our genomes in ways that can be passed on to other generations. We learn, and some of those ways in which we learn over time affect our neuronal connections throughout life. We're creatures of addiction, so that London taxi drivers have enlarged regions of certain parts of the hippocampus that's to do with learning and memory, because they need those mental maps of the streets of London that most of us don't need. And so on, those who have other skills have parts of the brain that are enhanced. And so our environment affects the way that we behave as much as our genes do. Genes provide the potential for our personhood, but they're not enough to define us and not sufficient to make us human. In fact, the human genome is something that supports development and learning and change with time. Just to show this not just taken out of the religious or Christian literature, here's a quote from the mainstream scientific journal Science. We're in the age of the genome, but we can still recognize that it takes much more than genes to make us human. Indeed, as Christians, we know that we live together and that community is extremely important. We're not defined just by our chemical makeup, but our relationships. I think it's an African phrase that says a person becomes a person through persons, our interaction with other people. Just as I said that DNA does nothing except in the context of a cell, so our genome does nothing to us except in the context of our interaction with other people. We could talk about, as if we talk bioethics, about stem cells. I think I'm going to skip past that and leave the medics to talk about stem cells at some other time. But what I would like to talk about for the moment is something emerging in biotechnology, and that is called gene editing. Can we modify the human genome? This was science fiction, really, until about, I'd say, 10 years ago. The idea that we could see what was something wrong with somebody's genome, where they had one of those debilitating diseases, and with a kind of cut and paste molecular scissors, chop something that was wrong out and put the right thing back in again. It was science fiction until 10 years ago, and now, within the last even 2 or 3 years, is now a real practical process. Let me introduce you to some of the techniques within this. TALENs is an abbreviation, and you see why it's called TALENs, because the full name is transcription activator-like effector nucleases. ZFNs, zinc finger nucleases, or now the technology that's used is called CRISPR-Cas9, clustered, regularly interspaced, short, crisp palindromic repeats. We'll call it CRISPR. It sounds a bit more like a cooking oil, I know, but it's better than the full name. These are methods whereby, using very precise molecular techniques, you can recognize a unique region in the genome and cut it out, either to debilitate it altogether or to replace it with something else. Gene correction tools. I won't go into any of the details in how it works, but let me give you some examples of way in which it's been used, at least in vitro. Infection with HIV. That retrovirus binds to receptors on the surface of the target cells. One of those receptors is the CCR5 receptor, and it was observed that a number of African prostitutes who were continually exposed to HIV did not ever get a disease. And looking at their genome, it's found that they have a mutation in the CCR5 gene. They cannot be infected with the virus, they don't have the receptor for it. So is it possible to knock out in healthy people that in their circulating blood cells, knock out that particular gene? It's on chromosome 3, and indeed using that sort of technology you can do that. You can generate cells in the laboratory that are totally resistant to HIV. Seems to have a medical benefit. Here's one that reached the press very recently. I think this is taken from a New Scientist. This young girl was suffering from an incurable form of leukemia and had gone through all sorts of kinds of chemotherapy and failed. And using cells that have been modified by this gene editing technology and given to her, she's in complete remission, having been close to death. A remarkable use of gene editing. But these are examples of what we might call somatic cells, affecting only that individual, not passed on from one generation to another one, changing their blood cells. What about germ cells and embryos? Something that would be passed on to the next generation. The genie was taken out of the bottle, in a sense, in April this year with a Chinese group who reported doing this with human embryos. Non-viable human embryos, and obviously not destined for implantation, but were able to induce mutations within the embryo that would then be carried on to the next generation. And without going into any of the technology of that, let's ask some of the questions. If we could do it, should we? And then I think it comes at different levels. Should we modify the genome of an embryo that would otherwise never be born? One that would suffer from an incurable genetic disease that would never reach term. That would be giving life where there never would be any chance of life in the first place. Should we modify the genome of an embryo that we know will go on to have cystic fibrosis? That's something that will manifest itself pretty much from the moment the child is born and will have a reduced lifespan. Should we modify the genome of somebody who has an inherited disease that will only manifest itself later in life. Huntington's disease, certain forms of breast cancer, where the person would live a normal life for 30 to 40 years. Should we modify the embryo there? And what about other more cosmetic means or reasons for modifying the genome? For changing eye colour or hair colour, whatever it might be, to make somebody look a little bit more beautiful. Scientists, I'm pleased to say, respond very strongly against the use of that technology at the moment. Partly because of risk, you might end up modifying wrong regions of the genome as well as the ones that you're intending to. So a number of publications in high-profile journals from very respectable secular scientists have suggested that we ought to be very cautious at this moment and not to do it. In fact, the research councils have issued statements on this. Wellcome Trust, Medical Research Council, the BBSRC. Active engagement with lots of stakeholders to discuss what we're doing before we do it. Francis Collins again, Director of the NIH. Advances in technologies that have given us an elegant new way of carrying out genome editing, but the strong arguments against engaging this activity remain. These include the serious and unquantifiable safety issues, ethical issues presented by altering the genome in a way that affects the next generation without their consent, and a current lack of compelling medical applications. So caution is necessary. So don't edit the human germline was the title of a letter to Nature earlier on this year. It's the slippery slope. It may seem to have good benefits to start with. It comes with lots of risk, and there are lots of other non-therapeutic applications it could be taken to. However, other theologians have been slightly less cautious and seen the positive aspects of this. Here is a professor of biology at a Christian college in the US. I'm not willing to write this work off as an attempt at playing God, he says. I think we would each play God every time we decide we would rather do things our way than God's way. When we put ourselves in charge of our health, our time and our resources, we put ourselves in God's position. Instead, this is an example of using the technologies God has given us in hope of reducing suffering. I can see redemption in this work. Now, I could go on for lots and lots, and we could talk about lots of other things, but I think at that point I will finish that. There are positive and there are negative aspects to genetic modification. The bioethics is very complicated, and hence I'm not going to give you the rights and the wrongs. You have to take the evidence and weigh that yourself. Thank you.
We have to finish by 4:30, the rules of the intensive, so we won't get into groups again, although in many ways it would be interesting for you to decide in your groups what the answers you think are to some of those. But I just want to give you the opportunity to either offer a comment, brief comment, or pose questions to Keith about anything that you've heard. Rachel's going to just beside you there.
Thank you. One question that is very controversial, obviously in the church these days, and does link into genetics, is the question of to what extent do sort of genes impact sexual orientation. I was just wondering if you could offer some thoughts from a scientific sort of research background on the relationship between those two.
Yeah, I guess that one might come up. It's, the answer is that it's not the way in which the media portray it. There clearly is some evidence of some genetic influence. The environment has a major influence on that as well. There is an extremely good article written by Dennis Alexander in Science and Christian Belief. I think there may even be copies on the piano, which go through the scientific evidence for whether genes, environment, etc. affect human sexuality. And rather than me explain that, I refer you to Dennis's article. If it's not on the one that's on the book table, I'm editor of Science and Christian Belief, and we thought that one was so important that it is freely downloadable to anybody. It's open access.
Just behind you, Rachel, to that.
I was wondering about the implications of some of the stuff that might be possible for how we view disability as a society. And I guess what sparked it was reading something the last couple of weeks about a fertility clinic allegedly screening out people with dyslexia and dyspraxia. So how would some of the stuff you're saying impact that kind of thing?
Thank you. And I 2 slides on from where I stopped was this, which is a quote from Tom Shakespeare, who lectures in the University of East Anglia, has a lot to say about disability rights. And his view on this technology is people with disabilities are, in my view, unlikely to be queuing up for genetic modification. Their priority is to combat discrimination and prejudice. And the way he puts it, the fixed genetic variations that cause a rare disease may seem an obvious act of beneficence, but such interventions assumes that there is robust consensus about the boundaries between normal variation and disability. The voices of people living with illness and impairment need to be heard. So yes, there is a kind of genetic discrimination that goes on in those cases. There are others who say that even if a disease is fairly debilitating, that we should retain it within the human gene pool rather than select it out. There are lots of these we know have, if they're there as heterozygotes, and there's one healthy and one diseased version of it, but there are benefits. Sickle cell anemia is an example. Those who are heterozygotes for that have increased resistance against malaria. And there are other examples of those sorts of things. And maybe we, at our peril, tinker with the long-term human gene pool because some of those mutations are there for advantageous processes. So yeah, thank you for that. Really good question. And we need to be very, very careful. This isn't a form of genetic discrimination against people. I go back to what I said close on the beginning, made in the image of God. We respect each person as who they are, not just on the basis of their genetics. It is that they're all individuals made in the image of God. Thank you.
I've got a question here at the front.
We heard this morning in history that people of faith were very much driving science forward. It now seems that we're the ones who are trying to push it back. How can we re-establish that thing that Christians and others of faith are actually moving science in a good way?
I think, that's a very good question, and partly that is very difficult. In the earlier days of science, a lot of the science was done by clergymen as a hobby almost. So they were engaged in it. The science itself was less focused and detailed and technological. Now it's such an expert area that you can't sort of indulge in it as a pastime. And there is a real danger, I think, of Christians appearing to be Luddites. Let's not change anything. We need to, I think, give credit and thanks where technologies can be used for benefit. But be there as voices who are saying, let's think about it before we use it. Not that it's necessarily wrong. This gene editing may not eventually be totally wrong, but let's pause for a moment. Let's stop and think through the implications for what we're doing. It's very easy to do a knee-jerk one way or the other, a knee-jerk that says, this is brilliant, we must do it, without thinking through the consequences. And the alternative knee-jerk is to say, well, there's so many problems with this, we should stop it now, cut. And I think it needs informed Christians to be involved in the debate. I am heartened that many of my scientist colleagues, especially with the gene editing, are there saying, let's stop, let's pause for a moment, as actually scientists did in the 1970s when genetic modification first came in. In 1973 there was a conference, I think it was Asaloma, where they put a pause on genetic modification while they thought through the implications for what introducing other bits of DNA into bacteria and plants and those things, what that could do. And sometimes we do need to be a stop and say, let's just think. Now the problem, of course, with technology and science now, it is so fast moving. It is such a competitive area. Now, I don't think Newton had great many competitors for Principia when he was publishing it. But nowadays, the drive to publish something before the competition gets in there is immense, and you can understand why. That Chinese group, they will be remembered forever as the first group who published genome editing of human embryos. It's interesting that that paper, so I'm told, was first declined by Science and Nature, the top scientific journals, because they questioned the ethical basis on which it was, the implications of that. And it's actually published in a lower ranking journal that it's not the obvious place to publish that work. So Christians should be engaged in the debate. They should be encouraging other scientists who are showing concern, and many are, to speak out.
We've got time for one last question. If anyone's got, yes.
The ethical basis on which we make decisions from a biblical or Christian perspective may not be the same, or they will not be the same as an atheist scientist. Is there any kind of coming together on consensus on what the basis of, or is it everything just taken on a case-by-case basis?
As a Christian, we would look to a higher authority for making our ethical decisions. But I think we would be able to say that that higher authority is not working just on a whim. There's usually a good, reasonable, rational basis why God has declared things should be in a certain way. And so human flourishing, looked at from a Christian perspective, it makes sense even to somebody coming from a non-Christian perspective. We can argue our ethics based on rights and flourishing. Clearly, in a secular context, to be able to say the Bible says, you're on a loser. But to take those biblical principles and to say God hasn't told us the way to behave just on a whim and because he felt, well, let's be nasty to people, but because that is the way in which society is best structured. This is the way in which people best behave. And I think we can make secular arguments for ethical principles that maybe are based on biblical ways of understanding, because it just makes sense. It's the right way to work.
Great, thank you, Keith, and thanks for giving us a run through something that has, I think, raised far more questions than it's answered, but lots of pause for thought. Thank you.
Thank you.